Welcome. Today's topic is central nervous system neurotransmitters. The contents of this topic will be the neurotransmitters, its properties, principles of chemical neurotransmission, types of neurotransmitters in the central nervous system, blood-brain barrier, and to summarize the neurotransmitters. So what are neurotransmitters? Neurotransmitters are a means for neurons to talk to one another. They act to facilitate, stimulate, or inhibit the postsynaptic neurons and the effector cells. There are over 50 to 100 known neurotransmitters in the human body. Neurotransmitters are endogenous chemicals which relay, amplify, and modulate the signals between a neuron and another cell. Neurotransmitters are packaged in synaptic vesicles that cluster beneath the membrane on the presynaptic side of the synapse and are released into the synaptic cleft where they bind to the receptors in the membrane on the postsynaptic side of the synapse. Release of neurotransmitters usually follows arrival of action potential at the synapse, but may follow graded electrical potentials. That is, low-level baseline release also occurs without electrical stimulation. Now, what are the properties of the neurotransmitters? Number one, they are synthesized in the presynaptic neuron, localized to vesicles in the presynaptic neuron. They are released from the presynaptic neuron under physiological conditions. They are rapidly removed from the synaptic cleft by uptake or degradation. Presence of receptor on the postsynaptic neuron. Binding to the receptor elicits a biological response. Now, there are several processes in the neurotransmitter action. They are, number one, the neurotransmitter molecules are synthesized from precursors under the influence of the enzymes. So they are synthesized from the precursors under the influence of the enzymes. Number two, they are molecules which are stored in the vesicles. After synthesis, they are stored in the vesicles. And these neurotransmitter molecules that leak from the vesicles are destroyed by the enzymes. They are destroyed by the enzymes. And the action potentials cause vesicles to fuse with the presynaptic membrane and release the neurotransmitter molecules bind the autoreceptors and inhibit the subsequent neurotransmitter release. Then the released neurotransmitter molecules bind to the postsynaptic receptors, and the released neurotransmitter molecules are deactivated either by a reuptake mechanism or enzymatic degradation. There's either a reuptake or there might be an enzymatic degradation of the neurotransmitters. Now, what are the principles of neurotransmission? It is the principles of chemical neurotransmission. Neurotransmitters are the chemical messengers that enable the neuron to neuron communication. Neurotransmitters are released from a presynaptic terminal and interact with the pre- and postsynaptic receptors. The inotropic receptors are directly coupled with ion channels, and the metabotropic receptors are coupled with G proteins and the second messengers. Now, the presynaptic receptors, they are also called as autoreceptors. They play an important role in modulation of the neurotransmitter release from the presynaptic membrane. And The activation of the presynaptic receptors in the CNS can result either by inhibition, that is muscarinic, alpha adrenoreceptors and dopaminergic receptors, or by enhancement of the respective neurotransmitter release, that is the nicotinic and adrenoreceptors. Now, what are the steps in the synaptic transmission? There are several steps in the synaptic transmission. Only 5 steps are there. Number 1, the synaptic transmission begins with generation of action potential in the presynaptic fiber to the synaptic terminal and activates the voltage signals and neurotransmitters are stored in the synaptic vesicle, released into the synaptic cleft and diffuse. Later on, these neurotransmitters bind to the receptors of the synaptic membrane. So these are bind to the receptors of the synaptic membrane. And after binding of the neurotransmitter, they cause a brief chain ionic conductance in the postsynaptic cell. So these are the various steps which are involved right from the generation of potential to the bind to the synaptic membrane. Binding of the neurotransmitter to the postsynaptic membrane causes a brief ionic conductance in the postsynaptic cell. Now, there's possibility. And this changes the strength from where the stimulus increases the strength of the stimulus, which increases so the threshold spike for generation is reached. Then, if you look at the type when threshold is given shocking, excitatory postsynaptic potential after this stimulus, where there's an inhibitory, actually excitatory, the inhibitory pressure after the spike threshold, which will cause the an inhibitory pathway prevents the excitatory potential from reaching the threshold. Now, if you look at these two graphs over here, one is the stimulation of excitatory pathway, and there is a stimulus, a supra-threshold stimulus given shortly after stimulating an inhibitory pathway, which prevents the excitatory potential from reaching the threshold. Now, what are the various types of neurotransmitters that are present in the central nervous system? There are 2 types of neurotransmitters, that is number 1, the excitatory neurotransmitters and the inhibitory neurotransmitters. The excitatory neurotransmitters are acetylcholine, aspartate, dopamine, histamine, norepinephrine, epinephrine, glutamate, and serotonin. Whereas the inhibitory neurotransmitters are GABA, gamma-aminobutyric acid, and glycine. Now, if you look at the drugs which are acting on the ANS, when I look into the excitatory neurotransmitters and inhibitory neurotransmitters. So when I talk about the excitatory neurotransmitter, a lot of cascade of changes which occur, and there is a release of neurotransmitter resulting in depolarization and excitation due to excitatory postsynaptic potential. Now, when I talk about inhibitory neurotransmitter potential, which causes inflammation. Now, neurotransmitters, neurotransmitters, that is a peripheral neurotransmitter, but the neurons are widely distributed in the central nervous system, typically in the cerebral cortex, reticular system, or semi-reticular formation, cerebellum, and the spinal cord. This is a structure of the acetylcholine. Then, contrarily, when you look into anticholinesterases such as tacrine, donepezil, and rivastigmine are used to improve the cognitive deficits related to Alzheimer's disease. The acetylcholine functions in the cerebral cortex, limbic system, and thalamus, and modulates arousal, respiration, motor activity, pain, and memory. The CNS responses to acetylcholine are mediated by muscarinic that is G protein coupled receptors, and nicotinic ligand gated ionic channel receptors. Now, when you look into the three mechanisms whereby binding of a neurotransmitter lead to a cellular effect. Number one, the receptors coupled to ionic channels. Examples are cholinergic nicotinic receptors and GABA receptors. So in this category, the receptors are coupled to the ionic channels, whereby the receptor which causes a change in the membrane potential or the ionic concentration within the cell and resulting in the intracellular effects. Number two, receptors which are occupied to adenylyl cyclase. The examples of this type of receptors which are occupied to the adenylyl cyclase, the examples are beta adrenoreceptors and alpha 2 adrenoreceptors, where the adenylyl cyclase converts the ATP to cyclic AMP, resulting in protein phosphorylation, and this leads to intracellular effects. Now, the third category is receptors occupied or receptors coupled to diacylglycerol and inositol triphosphate, DAG, IP3-DAG pathway. Receptors occupied or coupled to IP3-DAG pathway. The examples of this type of receptors coupled by the DAG pathway are alpha-1 adrenoreceptors and collagen receptors. Okay, resulting in intracellular. Choline is thought to modulate the arousal, it modulates the respiratory system, it modulates the motor activity, it is also associated with production of vertigo, it is associated with memory, and centrally acting antimuscarinic drugs are useful in managing or treating the Parkinson's disease also. Then, when you look into the synthesis and the release of acetylcholine from the cholinergic neuron, if you look at these steps, for example, synthesis of acetylcholine, it is mainly the choline is combined with acetyl-CoA to form acetylcholine, and the transport of choline is inhibited by hemicholinium. Now, this formed acetylcholine later on is uptaken into the storage vesicles, where acetylcholine is protected from degradation in the vesicles. So here the acetylcholine is put, or it is entering into the vesicles, which then is protected by degradation. So these synaptic vesicles later on, in the presence of calcium, okay, the release of neurotransmitter occurs. Okay, the release of neurotransmitter occurs, and this release is blocked by botulinum toxin, and spider venom causes release of acetylcholine. So here, the stored acetylcholine, which is stored in the synaptic vesicles, is released by spider venom, and it is blocked by botulinum toxin. Number 4, the released acetylcholine will bind to the postsynaptic receptor, which is activated by binding of the neurotransmitter, which will result in the intracellular response. Then, a bit of the acetylcholine, which is converted into either choline and acetate, or it is the acetylcholine, it may get degraded into synaptic cleft. The degradation of acetylcholine is rapidly hydrolyzed by cholinesterases into synaptic cleft. Later on, choline again it is utilized for the formation of acetylcholine. So this choline is recycled and to form again combined with acetyl-CoA to form acetylcholine. This is how it occurs. So this recycling of the choline mainly, which is the choline, is taken up by the neuron. Again, synaptic neuron, it takes up the choline. Okay, so this is how the synthesis and release of acetylcholine occurs at the neuron. Now, coming to the biogenic amines, that is the monoamines. In this monoamines, we have dopamine, noradrenaline or norepinephrine, and serotonin, which is also called as 5-hydroxytryptamine. All of these are neurotransmitters are monoamines, whereas epinephrine and histamine are also included in the monoamines. So the monoamines affect coordinated movement, they affect the behavior, mood and perception, learning, reward, sleep, and pain. So the monoamines, the biogenic amines, there are dopamine, norepinephrine, serotonin. And in addition to that, epinephrine and histamine are also included as monoamines. And these monoamines affect the coordinated movement, they affect the behavior, mood and perception, learning, reward, sleep, and pain. Now, what is dopamine? Dopamine is a neurotransmitter which is a biogenic amine, and the several drugs which affect dopamine levels are receptors in the central nervous system. There are 5 known dopamine receptors available that are grouped into D1-like and D2-like families. They are D1, D2, D3, D4, and D5. All dopamine receptors are metabotropic in nature. Now, dopamine is the most important of biogenic neurotransmitters in the central nervous system. The central dopaminergic pathways in the human brain are categorized according to the length of the neurons. The long projection of dopaminergic neurons are the most important part in the brain, which cover the substantia nigra, the ventral tegmental areas to the limbic cortex. Then, if you look at the structure of dopamine, so this is dopamine, and this is noradrenaline. So noradrenaline and dopamine. So dopamine is converted to noradrenaline by dopamine beta hydroxylase in the presence of oxygen, vitamin C, and copper. Now, the role of dopamine in Parkinson's disease. In Parkinson's disease, which is characterized by marked reduction in the concentration of dopamine in the basal ganglion, while in schizophrenia, there is dopaminergic overactivity in the mesolimbic, mesocortical, mesofrontal pathway, which controls the behavior. Dopamine is primarily an inhibitory neurotransmitter whose deficiency causes extrapyramidal disturbances. So dopamine, it is a primarily an inhibitory neurotransmitter. It is an inhibitory neurotransmitter whose deficiency causes extrapyramidal side effects. Now, if you look at the synthesis of dopamine, so dopamine, it is a catechol. So with catechol nucleus. So these are called as catecholamines, dopamine. So the primary precursor of dopamine is tyrosine. So tyrosine, in the presence of tyrosine hydroxylase, in the presence of oxygen, PHB, and ferrous, is converted into DOPA, that is dihydroxyphenylalanine. And this dihydroxyphenylalanine is converted to dopamine. Okay, and finally this DOPA is converted to dopamine. Now, what are these dopamine receptors? As I already told you, there are 5 types of dopamine receptors from D1 to D5. And dopamine is produced in several areas of the brain, including the substantia nigra. And this dopamine is also a neurohormone released by the hypothalamus. Its main function as a hormone is to inhibit the release of prolactin from the anterior lobe of pituitary. So dopamine is also a neurohormone released by the hypothalamus. Its main function as a hormone is to inhibit the release of prolactin from the anterior lobe of the pituitary. So, bromocriptine, which is mainly used in the treatment of prolactinemia. So, this dopamine can be supplied as a medication that acts on the sympathetic nervous system, producing effects such as increased heart rate and blood pressure. Since dopamine cannot cross the blood-brain barrier, dopamine given as a drug does not directly affect the central nervous system. So, to increase the amount of dopamine in the brain of the patients with diseases such as Parkinsonism and dopa-responsive dystonia, levodopa, the precursor can be given because it can cross the blood-brain barrier. Then, what are the functions of dopamine? Dopamine is having a central function to the reward system. Dopamine neurons are activated when an unexpected reward is presented. For example, dopamine is therefore believed by many to produce a teaching signal to the parts of the brain responsible for acquiring new motor sequences such as behaviors, movement, that is behaviors, movement, cognition, regulating prolactin secretion, motivation and pleasure like food, sex, and drugs. Disruption of the dopaminergic system, which occurs in Parkinsonism, schizophrenia, psychosis, and depression. When we talk about norepinephrine as a central nervous system and epinephrine as a central nervous system neurotransmitter, a few neurons produce norepinephrine, but these neurons innervate many regions of the brain. The receptors like alpha 1, alpha 2, and beta receptors are found in the central nervous system, which modulate the activity of norepinephrine. Now, what are these metabotropic receptors? They are metabotropic receptors. Primarily, norepinephrine is a neurotransmitter of postganglionic neurons of the sympathetic nervous system. In the CNS, norepinephrine is thought to modulate affective disorders like depression, learning memory, arousal, and pain perception. It is localized primarily in the cell bodies of the reticular formation, but the precise role of epinephrine in the CNS is still debatable. Now, how is this synthesis of norepinephrine from the adrenergic neurons occur? When you talk about the synthesis of norepinephrine, tyrosine is the primary source which along with which enters the cell, which enters the neuron in the presence of sodium is getting converted to dopa, and this dopa is converted to dopamine, and dopamine is converted to norepinephrine. Now, the synthesis of norepinephrine, which occurs with hydroxylation of tyrosine, which is a rate limiting step in the synthesis of all the other catecholamines. Then number 2, uptake into the storage vesicles. Dopamine enters the vesicles and is converted to norepinephrine. So this norepinephrine is protected from degradation in the vesicles, and the transport into the vesicle is inhibited by reserpine. Then the release of neurotransmitter, that is influx of calcium, causes fusion of vesicles with the cell membranes, and the release is blocked by guanethidine and beryllium. It is blocked by guanethidine and beryllium. Now, when you look into the alpha 2 receptors, so the alpha 2 receptors are mainly present in the presynaptic. Presynaptic, it is alpha 2. Postsynaptic is alpha 1. So in the presynaptic neuron, the alpha 2 receptors are present. The activation of this receptor decreases the production of cyclic AMP, leading to an inhibition of further release of norepinephrine from the neuron. Then the alpha-1 receptors, which are present in the postsynaptic neuron or postsynaptic cleft, the activation of these receptors increases the production of diacylglycerol and inositol triphosphate, leading to an increase in the intracellular calcium ions. Then if you look into the next neurotransmitter, which is 5-hydroxytryptamine or serotonin. The serotonin is produced by a relatively small number of neurons in the brain. These neurons project many regions of the brain. There are at least 12 different types of serotonin receptors. All these 5-HT receptors except 5-HT3 are metabotropic receptors. So all 5-HT3 receptors except all 5-HT receptors except 5-HT3 are metabotropic receptors. Now, the serotonergic neurons are found primarily near the midline raphe nuclei of the brainstem and project to the cortex, cerebellum, and the spinal cord. And this 5-hydroxytryptamine is thought to modulate the sensory pathways like nociception and homeostasis. Hence, the 5-HT3 receptor antagonist, which is used in the treatment of vomiting, nausea and vomiting. How is the serotonin synthesized? The primary amino acid, that is tryptophan, is converted to 5-hydroxytryptophan in the presence of tryptophan hydroxylase, which utilizes ferrous, oxygen, and THP. And this 5-hydroxytryptophan, again, it is converted to serotonin. Now, serotonergic systems play an important role in the schizophrenia, depression, temperature regulation, and eating disorders. 5-hydroxytryptamine is synthesized in the pineal gland, where it serves as a precursor for the synthesis of melatonin, presumably by an action within the hypothalamus. So, serotonergic systems, they play a very important role in certain diseases like schizophrenia, depression, the temperature regulation, and eating disorders. And 5-HT is also synthesized in the pineal gland, where it serves as the precursor for the synthesis of melatonin, presumably by an action within the hypothalamus. Then, serotonin is a monoamine neurotransmitter synthesized in serotonergic neurons in the central nervous system, as well as it is also synthesized in the GIT. The functions of serotonin are it regulates the anger, aggression, body temperature, mood, sleep, vomiting, sexuality, and appetite. The disorders which are associated with serotonin, that is, increase in the aggressiveness and angry behaviors like clinical depression, obsessive compulsive disorder, migraine, irritable bowel syndrome, tinnitus, fibromyalgia, bipolar disorders, and anxiety disorders are associated with serotonergic neurotransmitters. And extremely high levels of serotonin can have toxic and potentially fatal effects, causing a condition known as serotonin syndrome. In clinical practice, such toxic levels are essentially impossible to reach through an overdose of a single antidepressant drug. Now coming to histamine. So histamine and histaminergic neurons originate from the posterior hypothalamus. They project upwards to the cerebral cortex, to the limbic system, the caudate nucleus, the putamen, and the globus pallidus. Downwardly, the fibers projects include brainstem structures, the substantia nigra, the dorsal raphe, cerebellum, and the spinal cord. The histaminergic neurons play an important role in arousal, coupling neuronal activity with cerebral metabolism and neuroendocrine regulation. And also, it is helpful in regulation of food and water intake, the body temperature, the hormone release, and the sleep are the other roles of histamine. Then coming to the amino acids, we are having two types of amino acid neurotransmitters, excitatory neurotransmitters, inhibitory neurotransmitters. When you talk about the amino acids as neurotransmitters, excitatory include glutamic acid, aspartic acid, citric acid, and homocystic acid, which are involved in learning process, pain, and neuronal toxicity. When I talk about the inhibitory amino acid as neurotransmitters, they include GABA, glycine, taurine, and beta alanine. Now, excitatory neurotransmitter like glutamate. This is released into the synaptic cleft by calcium-dependent exocytosis. Glutamate acts on the postsynaptic receptors found on majority of the neurons. Not only that, there are different subtypes of inotropic and metabotropic glutamate receptors which are distributed in the brainstem regions. The glia take up the glutamate to terminate the action of the glutamate. GABA and glycine. GABA and glycine are inhibitory neurotransmitters. GABA is released by interneurons in the brain and the spinal cord, whereas glycine is released by interneurons only in the spinal cord. These are inotropic GABA A and metabotropic GABA B receptors. So, GABA A receptors are inotropic in nature, GABA B receptors are metabotropic in nature. Now, what are neuropeptides? Neuropeptides are produced by translation in the cell body, and the pre-proteins are processed in a tissue and the cell-specific manner to produce different peptides. There are many neuropeptides. So these neuropeptides can also co-localized with other transmitters to produce specific effects. So what are these neuropeptides? So neuropeptides and neurotransmitters may be released in a coordinated manner. For example, there are two subpopulations of neurons in the striatum. One population releases the GABA and enkephalin, and the other releases GABA and substance P. Okay, so these neuropeptides and neurotransmitters may be released in a coordinated fashion. Like, for example, the two subpopulations of neurons which are present in the striatum, one population releases GABA and enkephalins, and other population releases GABA and substance B. Now, coming to nitrous oxide and endocannabinoids. So this nitrous oxide and endocannabinoids are not classical neurotransmitters, but these compounds have profound effect on the neuronal function. Nitrous oxide is produced by an enzyme, nitric oxide synthase, which is activated by calcium calmodulin complex. I repeat, nitric oxide and endocannabinoids are not classic neurotransmitters, but these compounds have profound effect on the neuronal transmission. Nitric oxide is produced by an enzyme which is called as a nitric oxide synthase, which is activated by calcium calmodulin complex. And the drugs and endogenous brain lipids can activate the CB1 receptors and affect the memory, cognition, and pain perception. Now, what is the blood-brain barrier? The central nervous system is physically isolated by the blood-brain barrier, which prevent the entry of many substances in the brain. The blood-brain barrier is composed of modified endothelial cells, which with tight junctions surrounded by astrocytic processes. The drugs do not cross the blood-brain barrier, are highly lipophilic in nature. The drugs that do cross the blood-brain barrier are highly lipophilic in nature. And in early development, after injury or during infection, the blood-brain barrier gets compromised. Thank you.